So this is the next lecture in my Bio 410, Bio 510 Bioinformatics class. Today we're going to be talking about sort of pulling together how we've been talking about statistics and probability theory, all to better understand a specific bioinformatic application. And that's the application of sequence alignment. And so we're going to be pulling together a lot of the different ideas that we've talked about earlier, and a lot of what we were talking about, you'll see this is sort of the culmination of how we integrate those ideas. So it can be a somewhat complicated idea to understand the first time through, but just try to remember all the things that we've talked about already and how they're integrated together. We will go over some examples and show you guys how to actually do these kinds of calculations in class. But that's what the goal is today, is to try to integrate all these things to an actual applied case. So maybe it becomes a little bit less theoretical and a little bit more practical. So to do that, we're going to be focusing on the idea of Markov chains, and specifically we're going to be exploring how to use the steps involved in using a hidden Markov model in multiple sequence alignment. And through this process, I hope you guys will start to understand a little bit better how multiple sequence alignments are generated, and the sort of decisions and actions that go underneath it. I just want to point out this is only one method for doing multiple sequence alignments. It's not there are plenty of other methods, but it is an interesting and important one, and one that we actually use quite often in bioinformatics. So let's go back a little bit. So, we talked about random processes, stochastic processes, back a while ago when we were talking about distributions and sort of series of coin flips, et cetera, things of that sort. Today we're going to be talking about some more of these discrete random variables, right? So these discrete stochastic processes. And so remember, when we talk about stochastic processes, we're talking about something that's random, and we're going to be doing repeated observations. And so the kind of stochastic process we're going to talk about is a Markov chain. And that's a Markov chain is defined as a discrete time model that represents transitions of an object from one condition to another without memory, right? So it doesn't remember all the different changes that have gone on previously. In the case of a Markov chain, the probability state is only going to depend on what was the immediately prior state, not on the sequence of states that preceded it. So it's only going to compare about one step where you're coming from, right? One step behind. It's a great way to model systems that have a finite set of outcomes for each observation. And we're going to be using a specific approach that's known as a hidden Markov model, or an HMM. So the components that we're going to have n number of states, and if you can think of this way, they're going to be our nucleotide states, right? If we were thinking about sequence alignment, and we're going to have a state space that consists of the possibilities of these different states. We're going to have a random variable called x, which is going to describe the state of the chain at the ith observation. So we're going to have a series of observations, or a chain of observations. So x sub I is going to describe the state of the chain at position 1, or position 2, or position 3, etc. And then we're going to have some things that we're going to refer to as transition probabilities. And so these are the probabilities that you will move from one state to another state. So I'm going to move from, what's the probability? I'm going to move from state Si to state Sj. And so it's the probability Ij, right? So what's the probability I'm going to go from an A to a T, or from an A to a C, or an A to a G, et cetera, et cetera. So let's talk about sort of a non-sequence example, just to sort of get our minds around how this works. So suppose we have a molecule that moves between 4 cells arranged in a ring. And that molecule will remain in each cell for each time step with a probability of 0.9. It'll move to one of the 2 adjacent cells with an equal probability of 0.05. And what we're going to do first is sort of define what this transition matrix and state diagram we would associate with this problem. So if we have these 4 cells, right, we have this molecule, you could visualize it as a diagram like this, right? So we have position 1, position 2, position 3, and position 4. The molecule can either stay within each position with a probability of 0.9, or it can move to a neighboring position with a probability of 0.5 or 0.5. So it can't necessarily go across this way, as defined by this problem up here. So this is how we can sort of visually diagram out this state diagram of where this electron would be. But mathematically, we might present it like this. We'd present it as a matrix. So like in linear algebra, in the same way we would define I and J, as we've done previously in this class, I refers to the rows and J refers to the columns. So if I am in position 1, the probability of me going from position 1 to position 2, in this case, the P12 probability is 0.05, right? The probability of going from P1 to P3 is 0. It's an impossibility. And the probability of me going from P1 to P4, the P14 probability, right, is 0.5. And thus you could fill out this entire matrix, right, and propagate all the values. So we have this case, right, if we have the 4 cells that were arranged such that a molecule can move from any cell to any other cell with the same probability of remaining in the cell, how would this transition matrix change, right? What would be the thing that would indicate that change? So how would we make a change to this matrix? So right off the bat, you could consider which one of these, how this matrix would change, right? So if now I can suddenly go to any of these cells, specifically which number would have to change, which probability would have to change. Well, it's going to have to be this one, right? It's going to have to be that you would now add in the arrows that will allow me to go from 1 to 3, and from 3 to 1, and from 4 to 2, and from 2 to 4. And thus these 4 probabilities would have to change. I'll let you sort of guess and try to figure out exactly what that would look like. But the key question as we go, well, and we'll go over that in class, but it might be it would be a good exercise to see if you can do these calculations yourself. So what is the probability, leads us to the question of what is the probability of any specific path, right? This is kind of the idea that we're going to get to as we go through and think about sequence alignments. So what's the probability of any specific path? So if I know the starting and the ending position, can I actually calculate the probability of me, going from position 1 to position 3 in 4 steps, right? What are the different ways that I could get there? If we want to put that in sort of more explicit terms, when I'm talking about these paths, what I'm also really talking about is what is the probability that a system at state S sub I, so say in position 1, will move to position 3 in 4 steps, or 3 steps, or 2 steps, right? What is the probability of these other specific paths? And if I know these probabilities of either staying within my position or moving between my position, I can calculate these probabilities. So if we think about what's the probability of a specific path, what we want to do is we want to think about using our intersection probabilities, right? So if I think of, well, what is it going to take for me to go to P4 to P3? Right, and pretty straightforward calculation here, right? I can just say, well, the probability of me going from P4 to P3 is the probability of me being in position 4 and its intersection with the probability of being in position 3 at the next step, at position 1, at step 1. So if I'm at step 0 here, what's the probability I'm at step x1 in position 3 by the next step, number 1. And quite simply, if I know this probability is this intersection, right, by other equation, by our relationship and understanding of conditional probabilities, I can solve for this probability by making it equal to the probability of being in position 4 times the probability of being at position 3, given that at the previous step I was at position 4, right? So this is how I can actually generate what my probabilities, more explicit probability for this definition for this intersection probability. And what that leaves me with is, if I say I start at 4, my probability of being there is 1, right? And my probability of going to 4, 3 is basically going to be 0.05, right? So this probability, if I say what's the probability here, it's just 1 times this. So for 1 time step, it's a pretty straightforward way to think about this. So again, this is the same sort of structure, right, that we talked about, where we had the probability of the hypothesis given the data equals the probability of the intersection of the hypothesis and the data divided by the probability of the data, right? This is the same kind of equation that we've seen previously. Now, what if we think of a slightly more complicated example, right? Now, what if we're thinking of the case where we might say we've got a path where we're going to sort of start at 4, go to 3, and go to 4. How would we then calculate this probability? Well, if we're going to calculate the probability of going from 3 to, if we're going to calculate this probability based upon this previous example, right, we're going to say, well, it's the probability of going from 3 to 4 given the probability of being in the 4 position at step 2, right? And so now, instead of being what's the probability being of x being at the 4 position in the first step, we have the probability of what's the probability of going from 4 to 3, which we know over here, right? And now we're going to multiply that. We're going to solve the equation the same way we did up here, but we're just going to substitute in these other entities. So now we're going to solve it for the probability of going from of being at step 2 in 4, given that in the previous step we went from 4 to 3. Again, now we have to calculate this example, right? So we know from above that this probability is p 4, 3 position, 0.05. Now we've got to figure out this equation, right? This probability right here is somewhat complicated, right? We have to figure out what's the probability that we're going to go, we have to include, derive out this example, right? So in this case, what's the probability that we're going to be in 4? And we're going to substitute in this union again, right? I can then rederive out this example, and now I've got the probability that I'm going to be in the 4 position at the second step, given the probability that I was in the first position at the third. I was in the 3rd position at the first step. And all that goes to say is, it just goes to basically multiplying these probabilities together. So this is how we derive this example, but I think the main lesson to learn here is that in the end, because of these definitions that we were able to learn earlier about the relationships between these things based on the fact that they're conditional probabilities, right? We're able to figure out that the probability is actually going to be of going from 4 to 3 and then 3 back to 4 is going to be solved by a multiplicative principle, right? And that's the probability of going 4 to 3 times the probability of going back to 3 to 4. You know, that multiplicative principle is pretty important. Now, you might want to think about, how, remember, this is an example of how we might think about independent probabilities. These are examples of how we might think about dependent probabilities. But what really is important here is that we understand that the probability here is just going to depend on the most recent state of the model, right? And this is what is referred to as the Markov property. So even if you have a whole bunch of multiple states, it's only going to depend on what the previous state was. And that's sort of the process. This lack of memory of the system is how the system actually maintains its independence. and is able to multiply itself out like this. So as we think about like what are the probabilities of me going from 4 to 3, then 3 to 4, then staying in 4, then going from 4 to 1 and staying in 1, and then going to 2. So the probability of this specific path, right, through this diagram, so 4 to 3, then back to 4, then stay in 4, then 4 to 1, then stay in 1, then end up in 2, is these series of probabilities, right? So in this case, that would be the probability of going from 4 to 3, right? Times the probability of going from 3 to 4, times the probability of staying in 4, times the probability of going 4 to 1, times the probability of staying within 1, times the probability of going 1 to 2. And that's how we can calculate this probability. So another way to think about this, right? This is when I talk about the idea of what is the probability of moving from that a system in state 1 will move to a state J in so many steps. I have to understand that like I'm not only thinking about one case and one pathway through, right? When I think about this, what's the probability of a system, right? I have to think of all the possible paths that I could get from, say, 4 to 2 in 1, 2, 3, 4, 5, 6 steps. And so if I'm going to figure out the probability of a system from getting there, or a model for getting there, right, what I'm actually going to do is I'm going to use this equation. And there is a lot of sort of linear algebra involved in this, and I'm going to kind of gloss over some of these examples of like how linear algebra works. We'll talk a little bit more about that in class. But I just want you to see it's not really, you know, I think it's helpful and it's useful and it's important to know the mathematical theory that sort of underlies what we're doing. But basically, I want to show you how it's practically applied, because I think it becomes a little clearer and intuitive when we look at it that way. So if I'm going to actually figure out what this other probability is here, right, what happens is we're going to have this k-step transition probability, and we're going to calculate that from a matrix that we're going to call P of k, right? So in this case, if I have P of 2, right, and this is the system that I have, right, what's the probability that I'm going to go to the next step given this probability matrix of probabilities, transition probabilities, right? What I'm going to have is I'm going to actually do for 2 steps. I'm going to be able to generate a I'm going to do some matrix multiplication. And so when I do that, like quite simply, it's easy enough for me to think of when I multiply these 2 matrices together, I end up with something like this, right? And so I'll go over sort of a more detailed process by how we do matrix multiplication, but just know that we're multiplying these 2 matrices together. And what that means is then if I want to figure out how do I go from position 1 to position 4 in 2 steps, right? I basically have to calculate all the different pathways that I could possibly use. And to do that, right, I'm going to look at from position 1 to position, I'm going to stay in position 1 and go to position 4, right? And that's going to be 0.09 times 0.5, right? So 0.09 times 0.05, right? Or I could go to position 2, right? And then go 2 to 4, right? So I'd have to have a slightly different, that's impossible, I can't do that. But again, I have to add that to my path, right? So position 2 to position 4, right? Times, so I have to multiply that. So what's the probability of going from position 1 to position 2, and the times position 2 to position 4. The other way I could do it is if I was in position 1, I could go to 3 and then 3 to 4, right? And then I could go from position 1 to position 4 and then stay in 4. So those are my 4 possible outcomes, right? If we think about the probabilities associated with those possible outcomes, right? We got 0.09 of staying in 1 and then going to 4 is 0.5. So that'd be those two multiplied together. If I was to think of the probability of going from 1 to 2 and 2 to 4, well, 1 to 2 is 0.5, but 2 to 4 is a 0. It's never going to happen. It's not part of the model. Same thing with going from 1 to 3 and 3 to 4, right? Going from 1 to 3 is 0, but going from 3 to 4 is 0.5. So that's also going to be equal to 0. And then if I think of going from 1 to 4 and then staying within 4, right? That's 0.5 times 0.9. And if I calculate this out, I end up with a probability of 0.9, right? So what's the probability of me going from 1 to 4 in 2 steps is 0.9. And that's how this sort of would play out for each one of these positions, and you would calculate this basically for each of these. Now, there are functions that allow you to do this, but we don't have to add this up. But I think what the most important thing I want you guys to see here is that I'm adding them up, right? I'm calculating each of these individual probabilities of these paths, and I'm adding them up into one total probability, right? So this is looking at disjoint unions. And this is the additive principle that we talked about before in calculating probability. So I have to calculate all the probability paths. Think of this like calculating the marginal likelihoods in the Bayesian examples that we've used before, right? Where we have to calculate all the possible, like the total probability that we have at the bottom is going to be added up by calculating all the possible outcomes. Right? So this is that same idea. I have to calculate all the probability associated with any of these paths and then add it up, and that's what's going to go in my value. So if we have this case, take a second, see if you can figure out how you would calculate if this was a 3-step process, right? And you were starting at position 3, right? And going to cell 2 in 3 steps. What is the probability of that path? What is the probability of that system starting at position 3 and getting to position 2 in k number of steps? See if you can calculate it out. That's kind of like a little dummy example of how the Markov model works and how to sort of build the state diagrams and associate the appropriate matrices of transition probabilities with it. But we're going to be looking at something known as a hidden Markov model. And the hidden Markov model is a probabilistic model that assigns likelihoods to all possible combinations of gaps and mismatches that are determined to be most likely in a multiple sequence alignment or set of multiple sequence alignments. And basically in the same way before, where we were looking to describe a system, so all the possible outcomes, instead of a single outcome per state, right, each state is going to produce several possible outcomes. So there's going to be probabilities of all those paths, right? And we're trying to figure out what is the probability of that specific movement from one state to the next state. And we could use another example here in the case of a fair coin, right? So we had this previous example where we had to, we were picking a coin at the beginning and we had to decide if it was fair or biased based upon a toss of an outcome, right? And so you either got a fair coin or a biased coin, an F or a B. And each state you flip the coin a bunch of times, and then you would base you would make a hypothesis based upon the number of flips. So the hypothesis given the data, right? And if we think about how something like this might look, we might look at this case, right, where we have a fair coin, right, and a biased coin. We've got a 50% chance of picking either one, right? Once we pick one, we stick with it. We flip it a bunch of times, and we have a biased coin that might have a 2/3 probability of getting heads and 1/3 probability of getting a tails. And then we flip the coin a bunch of times, and after a bunch of flips, we can figure out whether or not we think we got either a fair or biased coin. That's one possible model that we could generate, right? That we flip it and they can't talk back and forth. There's no change, right? We could also structure this example a little bit differently. So instead of, I could choose a model where each time I flip a coin, I pick a new coin. So I pick either a fair or biased coin, I flip the coin, I get a result, right? And I would either have a biased or a fair coin in that regard. I also could say I could base it upon the coin that I picked, right? So this is me just picking an extra coin. I also could just say, like, I'm going to flip this coin, and then depending upon if I get heads or tails, I'll go over here, and I'll flip this coin, and depending upon heads or tails, I'll go over here, right? And so we can start to see that we have different structures, different models that are going on here. And the thing that's important to remember about the hidden Markov model is that I'm not going to ever know what if I picked a fair or biased coin per se, right? The data that I'm going to be dealing with are the heads and the tails, right? And I'm trying to make a distinction and test the hypothesis about the state of this unknown thing that I did. Did I pick a fair coin or did I pick a biased coin? Is it this? Does this model best fit or does this model best fit, right? And what you start doing that is you start seeing that you have these unobserved states that are involved in this process, right? I don't know what I don't know what all the outcomes are that are in between. And that's what's going to happen when we start doing hidden Markov models, is that as we go through and we flip these coins, right, we might only know what is the first and last flip, or maybe we know what a series of flips might look like. But we don't necessarily know what all the stuff in between is. And in that way, I think that this system, you can start to understand why it's important for modeling sequence probabilities or sequence alignments. Because, because of evolution happening over time, we don't necessarily know all the intermediate states of a lot of these systems, right? We don't know what the ancestral state is per se of a sequence, or what it was 10 generations ago, or 20 generations ago, or 1,000 generations ago. And so we have to deal with a lot of unobserved states, but we do have an outcome, right? We do have sequences that come from outcomes that we can use to model these things. So here's an example of a state diagram for an HMM for a sequence, showing where you might have a sequence that switches between, say, a normal region and a GCC-rich region, right? And so I think one of the things I want to point out to you here is that we have these different states. We define the movement between these different possible whether or not you're in a normal region or a GC region. We refer to these as transition probabilities, and we refer to the state within each position using emission probabilities. So notice that depending upon whether you're in a normal region or a GC-rich region, right, the probability that you'll be an A, a C, a G, or a T changes depending upon which state you're in, right? Which type of region you're in. And using this kind of structure that we have here, we could say something like, is a TGCA in a GC-rich region of a genome? So if I sampled TCGA, if that was my data, and I have a hypothesis, what's the probability that it's in a GC region, right? If I'm going to test this probability, again, I can use this same kind of probability structure, where before I just had I would have had the probability over here, right? This is the exact same one that we saw back in our previous slides here, right, where we had the probability of the intersection of two entities times the probability of one times the probability of the other given the one, right? Same sort of structure, just slightly, now we're solving for this conditional probability, right? So this is just the intersection of the probability of TCGA intersecting with GC-rich divided by the probability of TGCA. And so this is the total disjoint union of all the probabilities of TGCA. So if we think about like one of the possible paths that we could get through this diagram, right? We could go T, C, G, A, right? And so in this case, we would start, T, we could have T in a normal region, then we could go a C in a GC rich region, and a G in a GC rich region, and to an A in a normal region, right? That's how we could move through this. And if we're going to move through this, right, we can see that we have these different probabilities of transition across that associated with the path, as well as emission based upon the likelihood of observing a specific type of nucleotide in those positions. So if we were to think about how we would calculate what's the probability of a path, right, of NRRN, right, and so that's seeing that we have this sequence in a GC-rich region, given that intersecting with the sequence of TCGA, right? We calculate that by calculating each of these probabilities multiplied to each other. Right? So we've got the probability of the start times the probability that we have a T in a normal region times the probability of going from normal to GC rich times the probability of being a C in a GC rich region times the probability of staying within a rich region, 0.75, times the probability of being a G in a rich region, times the probability of going from a rich region to a normal region, times the probability of being an A in a normal region, times the probability of ending the sequence. And so if I'm going to calculate out that probability, right, it's just 0.5 right here, 0.25, 0.25, 0.35, 0.75 times 0.35 times 0.25 times 0.25 times 1 right here. So the probability, in other words, that I am going through a normal, rich, normal region intersecting with this specific sequence is 1.799 times 10 to the fourth. Now, if I actually want to figure out the probability of observing a TC-rich region coming from a GC-rich region, right, I have to look at a lot of other paths. I can't just look at this one path. I have to look at a whole bunch of other paths. And I have to figure out what's the probability, what's the proportion of the probability of TCGA that intersects with going through GCC-rich. Right? And so I will calculate the probability of each of these paths independently, right? Each of these possible combinations. And there's a couple questions you could ask for, right? In this case, how many possible paths are there? We've gone over on how to calculate that. I wonder if you can figure it out. What is the sum of all the probability paths what is the sum of all the sequence path probabilities that are for TCGA. So what's the total probability down here? What does that look like to you? That looks like something we're going to have to calculate using the additive principle, right? We're going to have to add up the values for each of these probabilities to get the total probability that's going to go here, right? Looks also kind of familiar. We might think of that as being like our marginal likelihood value. Right? And then if I'm going to figure out what is the probability associated with TCGA intersecting with GC-rich using all these different paths, right? That's just looking at the proportion of the total that is associated with going through a GC-rich region. And so the answer to this question is approximately 38%. See if you can calculate it for yourself. It'll be a good little test to see if you can actually do the calculations yourself. I will say, this example is well covered in the book, chapter 12, in the section on stochastic processes. So if you look at concepts in bioinformatics and genomics, the Momand and McCurdy book, it's very well covered there. But see if you can do these calculations yourself. So I think it'd be good practice for you. So that's one example of how we might move through these hidden Markov models. I want to specifically talk about how we can apply them to look at sequence alignment. And so we're going to be doing hidden Markov models in multiple sequence alignment. And these models actually, they originally came from language analysis, which is pretty cool. But we're going to be using them to try to identify common underlying structures from different species. So in other words, can we find sequences that are similar to other sequences of this type? So we might look for things that have the same type of sequences. So maybe like conserved domains of proteins and things like that, because they might have some similar function or shared evolutionary history or something of that sort. So the kind of profile that we're going to be looking for here is going to be defined by 3 states. We're going to have an insertion, deletion, or match in this case. That's the states that we're going to be looking for. So an insertion, deletion, or match. And each insertion and match state has an emission probability, and for each nucleotide identity that we might see. A deletion only has one outcome, right? It only has to introduce a gap. It's not going to allow for, since there's no nucleotide in that position, we wouldn't have any emission probability associated with it. And we can do things like this. So we could look to say maybe use this kind of state diagram, to do things like, generate what the ancestral sequence is of a specific topic or something. And the idea here is that if we think about things over evolutionary time, we would expect for them to be divergent from each other, but that they might maintain or still fit this same model that we're going to be generating. So we might think that, over evolutionary time, that there might be some sequence change. But if we start from here, we could calculate what is the probability of going from M1 to having a deletion as the second nucleotide, to then having a new, or going to a match at M3, right? Then M4, and then adding in a new sequence right at the end. We can figure out what the probability of this path is, right? Or what this hypothesis is as part of the possible outcomes. So if we think about how we apply this approach to multiple sequence alignment, usually what happens is we have a set of DNA sequences that we've identified that produces a common protein structure or function. And so we get a set of initial sequences that we call our training set. So ones that we know are associated with the thing we're looking at. And after that, we would then set the model size, or the number of states, and any preliminary estimates we have for transition and emission probabilities. So usually we might have some information. I'll show you in a second how we might go about calculating that. But we could do an initial alignment and figure out how to calculate some of these probabilities. Then once we have those probabilities in place, we might actually go through and do some optimization to maximize the chance that if we add in something random, we'll actually be able to generate one of the, if we generated, so once we have that in place, once we have that model in place, what we would do is then go through and optimize the probabilities. And what that means is that we're going to try to maximize the chance that if I was just to generate something random out of that model, that it would actually be one of the training sequences. And this is called training the model. So I'm going to train the model so it reproduces the actual data that I initially started with it. Once we have a trained model, then we can align new sequences or test sequences that were not in the training set to figure out whether or not there's some sort of common structure or function that's present in these new sequences that we see that's related to this model. And so for each new sequence that we align, we can do something that's called generating a likelihood. And that likelihood is the likelihood that the random output of the trained model would be the new sequence. So is it likely that what we're looking at is random, or is it likely that we're looking at as a good match? And so this probability is then interpreted as the confidence of how the new sequence is evolutionarily related to the training sequence. So kind of the initial steps that are involved in this process is we build this initial model. So in this case, we're going to have, and I think, you know, the key thing to keep in mind is the model that we're going to be building is going to look something like this, right? It's going to be looking something where it has insertion, deletions, and match states, right? This is the kind of model that we're going to be building. So the first thing is we need a training set. So say we were looking at something like TP53 promoters, right? We're going to have our model size. Remember that we said one of the first things that we're going to do is set the model size. So first we get our sequences, then we set the model size. So we set the model size. We see that we have rats, sheep, pig, human, and hamster as our data set. And then we can begin to set the model size. And so in this case, we'll have possible matches for points 11 through 18. We'll have possible deletions from 11 to 18. We'll have possible insert at 18, and then some matches and deletions from 19 to 24. So once we have these, we can then go through and calculate the preliminary transition and emission probabilities, which are based upon this set. So if I had to say, like, you know, what's the emission probability that I'm going to be a C at this first position, right? I would look at these, if it's a match, I would look at these 4 possible probabilities and say, well, it's 75% chance I'm going to be a C at this position, and 25% I'm going to be a T. Right? If I'm a match, there's also a probability that, well, what's the probability that I'm going to be a match or a deletion? Here I could say, well, the probability I'm going to be a match or deletion is 4 out of 5, so 80%. The probability I'm going to be a deletion is 20. So in that way, I can set these preliminary values in this training set. And so what happens is over time, I'm going to train this model to create an optimized model. So what that does is I'm going to look and generate a likelihood of each model. The likelihood of the model is just going to be continually maximized so that the product of the sequence probabilities over all training sequences are maximized. So again, just to remind you, we're going to be dealing with a model that's going to be looking something like this, right? Where we're going to start with, it's going to be made up of match states, insert states, and delete states. So before I talked about how we would generate this maximized value of this overall likelihood, what that actually looks like is we're going to apply something called the BombWelsh algorithm. And in that specific algorithm, we're going to be looking for the locally best model that we can find after generating these probabilities from our initial model. And basically the idea here, right, is we're going to take the probability of a specific training. We're going to try to figure out what the probability is of a specific training sequence given a model. And what that is that's the sum of all the intersections of that sequence with paths given that model. And we're going to sum up all those paths. Again, this is that total probability, right? And we're going to sum it all up, and that's going to give us, you know, the probability of this one specific sequence. But then we're going to look to see, well, what's the probability if I give it a set of sequences, right? And again, you're using your training set to do this. So you're trying to figure out what's the probability if I give it a set of sequences that I'm going to get a set of sequences given the model, right? And this is this maximum. This is the likelihood of the model over here. Right, the product of the sequence probabilities of all the training sequences. And the idea here is that you create a model, you use this probability structure to create a model which will, at random, always generate something that looks like one of your training sequences. So that's the idea. We're going to generate the model that's the most likely to produce something that looks like your training sequences. So it has the specific steps, right, where it's going to, you know, assign the initial probabilities to the model, and then it's going to go through and it's going to compute new transmission and emission probabilities following these 2 rules. And these rules are rather complex, but if we just sort of look at one of them, right, it's going to add up all the sequence path probabilities for a given transition. So all the possible transition probabilities for this going from, say, like I to the M position, right? And then once it adds them all up, that's going to be the new probability for going from I to M. And then once it has added up all these new probabilities, then it's going to rescale everything to 1, right? So it's going to rescale all the probabilities that are coming out from I to M and make sure that they are rescaled to 1. This causes the highest probability of producing the training sequence by giving greater weight and transitions to those taken by the path which has sort of the most likely outcome. And it'll, in addition to that, it'll add up after it does that. It'll also add up all the sequence probabilities that emit particular nucleotides at, say, position M3 as well, and do the same thing for the emission probabilities. So it does it for the transmission probabilities, then does it for the emission probabilities, and then it replaces them all, and you get a new model, right? And then you're going to repeat the entire set again, right? Each time improving the model and its ability to look more and more like your training sequences. So eventually that model will produce will be able to produce at random something that looks like your training sequences. And that's the learning process. It's a rather complicated algorithm to show you how it works, and there's definitely some challenges in iterating through this. But you might run this thing for about 10 iterations. And again, it's a model that looks something like this, and we're just trying to look at, what is the probability in this one case. So after we've created this trained optimized model by maximizing the likelihood function over here, then what we can actually do is add new sequences to it. So we can add new sequences to the multiple sequence alignment. And basically what we do is when we add a new sequence in, we use this algorithm called the Viterbi algorithm, which is basically just a way to find the most likely path through the model for this new sequence. So given that model, what's the best path through that model? And we call that the Viterbi probability. And based off of that probability, we can then calculate how distant is the sequence that we added to it from the model. And this is basically referred to as a goodness, or actually a badness of fit measure. But we can see, is it very distant? Is it very close, right? And we're looking for something that is very close to that model. And then by this way, we can look and see if it's a good match to this learned model. So that's how we think about an application of the hidden Markov model. There's a few examples in this how we might apply these sorts of principles. You can see the intersection and integration of a whole bunch of the probability concepts that we've talked about in class: additive, multiplicative approaches to probability, conditional probabilities, Bayesian approaches. Basically, this initial training, when we calculate these preliminary transitions and these emission probabilities, those are Bayesian priors, right? These are Bayesian priors. And then when we go through this bomb and wash algorithm, as we are constantly updating these probability structures, that's like how we would update with prior information. Every time we do an iteration, that prior information will change. Right, so this is an example of how we could apply these things to this sort of discrete stochastic case, stochastic process, and we'll go over some more examples in class to make sure you have a better understanding of it. But thanks for paying attention.